Hello, everyone, and welcome to today's webinar, Label-Free Characterization of Challenging Transmembrane Proteins Using Alto Digital SPR. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Nicoya. To learn more about our sponsor, please visit nicoyalife.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would now like to welcome our speakers, Dr. Michael Piazza, Director of Applications Development, Nicoya, and Dr. Anil Kumar, Head of CMC in Europe, ACROBiosystems Group. For complete biographies on our speaker, please visit the Biography tab at the top of the screen. Mike and Neil, welcome. Mike, kick us off.
Thank you so much, and thank you for everyone for joining us today. Hopefully you enjoyed this talk, and yeah, if there's any questions, please let us know, or you can contact us after also. So for today's presentation, I'm going to start off by giving you a brief introduction into SPR, followed by a background on our company Nicoya and our story and how we got to where we are. And then I'll introduce you to our Alto platform and its unique capabilities, followed by some binding kinetics of challenging transmembrane proteins. So we'll start first by going through some basic SPR theory. Surface plasmon resonance, or SPR, is a label-free optical method of measuring the binding interactions between two partners. This diagram on the left shows the basic assay format that's used in SPR. This consists of a gold sensor surface that performs the detection for us, followed by the immobilization of one of the binding partners to the sensor surface, which is referred to as the ligand, while the other binding partner, referred to as the analyte, is going to be free-flowing in solution. The binding, which is measured in real time, is directly due to a change in the optical mass. The property of our sensor that we're going to be monitoring is called the absorbance peak. The gold is going to absorb light, creating this absorbance peak, as shown on the right-hand side. The center position of this peak is going to shift as binding events. In this case here, the analyte binding to the ligand occurs, and the new peak position will give us our sensor response. We then collect these and use these to determine our binding kinetics. A typical sensorgram includes a baseline phase where buffer is passed over the immobilized surface, followed by the interaction of the analyte to the ligand, referred to as the association phase, where we measure the initial binding to the ligand. Next, we then pass a buffer drop over the sensor to collect data for the dissociation phase. In this situation, the analyte is dissociating from the immobilized ligand. In cases where this dissociation is too slow to go to completion, we have to use a regeneration step to remove this analyte from the sensor. Now, why do we want to use SPR? What are the uses and benefits of it? So SPR can be something as simple as providing yes or no on binding data. However, the real workhorse of this platform is obtaining kinetics data. So this would be the association and dissociation rate constants, as well as the equilibrium dissociation constant, typically referred to as the affinity. We can also use SPR to do quantitation of unknown concentration determination. We can use it for epitope mapping and binning, or simply screening a large panel of compounds. And there are lots of benefits to SPR, namely it being label-free, highly specific to the target you're looking for, real-time data, and low sample consumption. Next, here, I'm going to go into the intro to our story at Nicoya. So our mission is to improve human life by helping scientists succeed. And we've been working on this mission since our CEO, Ryan, started the company in 2012. At that time, he was frustrated with the lack of accessible and affordable instruments for label-free analysis, and thus spent 5 years developing a better solution, our first OpenSPR. OpenSPR at the time was the world's first benchtime instrument for SPR analysis, and since its launch in 2015, has democratized this technique to help hundreds of scientists get access to this powerful data to help accelerate their missions. This has motivated us to develop the Alto, which is the world's first digital SPR system. We, as a company, are a team focused on supporting our over 500 customers across 50 countries constantly pushing the limits of technological innovation. This has resulted in over 200 patents filed on our products and has allowed our customers to publish in over 250 peer-reviewed journal articles, spanning all disease types and therapeutic approaches. We have been accomplishing this by helping scientists simplify their research with our benchtop open SPR and digital SPR platforms. So what do these platforms do and how do they work? I'll go into an Alto overview here. So after having these thousands of conversations with scientists, we realized that while SPR remained a critical technology in every lab, the solutions that existed made scientists compromise on their workflows and their projects. And if you're familiar with any traditional SPR BLI, these challenges will be all too familiar to you and really tie back to traditional fluidic systems. So Alto, as the world's only surface plasmon resonance system powered by digital microfluidics, or DMF, has revolutionized real-time interaction analysis by taking this complexity out of the SPR and eliminating the need to compromise on quality and time. The Alto platform consists of a fluidics-free benchtop instrument, shown on the left-hand side here, which has a touchscreen-enabled built-in PC, which is super compact and does not require a connected PC, so you'll only need about 1.5 feet of benchtop space to use it. The instrument itself contains the electronics, optics, and temperature control elements. Well, we took all of the complex traditional fluidics found in other instruments out of the actual instrument and integrated them into a disposable cartridge. This allows for the movement of fluidics, has the SPR sensors integrated into it, and the storage for the samples and reagents all in one place. By using this DMF, we're able to move discrete nanoliter volumes of sample throughout the cartridge by the application of voltage to individual electrodes. And then lastly, our cloud-based design and analysis software lets your data be accessible anywhere you are, so you can check in on your results and design your experiments before you even enter the lab. So what are the benefits of this system? How does this affect you? Well, for starters, you'll only need 2 microliters of sample for a full kinetic evaluation. That is about 100 times less than conventional instruments. This means you don't have to worry about wasting sample that took you weeks or months to produce, and you can start using SPR earlier in your discovery process to find more hits or more candidates. From these 2 microliters of sample, because of all the work that is done on the fluidic side in the cartridge, you'll still get your amazing high-quality data with minimal hands-on time needed. This is because the Alto is designed to save your precious resource, which is mostly your time and your team's time. This is done through its automated onboard serial dilutions, ability to handle crude samples without purification, since there's no pumps or tubes to be clogged, no need for degassing, cleaning, or labeling, and its simple cloud-based design and analysis, which can save your team up to 70% of time compared to other solutions. And now, because all of the fluidics are in the cartridge, the instrument is very robust and reliable, meaning there's no downtime, no maintenance, or service contracts. The simplicity in hardware is critical for these instruments, since we do have 16 channels, which means that there is potential for a lot to go wrong. However, because of its robust nature, this is very unlikely. The instrument is also very compact, meaning you can put it anywhere in your lab. And lastly, traditional instruments have limited protocol flexibility because of the fluidic pathways and hardware is fixed. With Alto, we've completely changed the fluidic design with a simple software update, meaning that protocols can continue to expand and improve without the need to buy a new instrument. Now, while we've added some of these also awesome features and capabilities by leveraging our cloud-based tools, the workflow is going to be super familiar. You design an assay in a simple 3-step process, following the same assay steps you would in a traditional SPR or BLI experiment. And then you use the instrument's built-in PC to set up and run and acquire the data. And then our analysis software will automatically fit your data with the tools that you need. Now, digital microfluidics, the DMF, what is this and how does it work? This is the ability to automate the movement of your sample drops on a cartridge via electrowetting to allow for the traditional protocols and processes of immobilizing the ligand, interacting your analytes, and regenerating to start your analysis over again. So taking a closer look at the Alto cartridge, we can see how these samples are moved. This is achieved by the application of voltage to a grid of electrodes. By turning these electrodes on and off in sequence, we can move these samples throughout the cartridge. In this example here, we see liquid coming from a storage well being transported up towards the SPR sensors. Now, although Alto only needs 2 microliters, it still produces very high quality data. This pump-free design ensures that there is 0 concentration dispersion between your analyte sample and the buffer. There is also no sample evaporation, ensuring that you always get accurate kinetics, regardless of interaction times. And you can also maximize reproducibility with the automation of these tedious steps, like serial dilutions and purifications. Here we can see how these serial dilutions work on the Alto cartridge. Dispensed buffer and sample meet in the mixing zone. So in these examples here, you can see the brightly colored drops getting more faint as they're mixing with the buffer to produce these serial dilutions. These occur independently in each lane, allowing for up to 8 sets of 5 diluted concentrations per lane of interaction. And then finally, each of these 350 nanoliter droplets are then delivered to the sensor surface. In this example here, we can see the droplets oscillating over the sensor. The sensor is basically in the middle of the drops that are oscillating right now. In this sequence, we can see the green colored drop acting as the association phase, allowing us to collect the binding interaction data for our analyte, followed by a clear buffer drop to collect the dissociation data. All of this happens automated without the need of having to program any of these interactions. So as a summary of the features for the Alto instrument, we feature a 16-channel cartridge powered by this DMF. It has a maximum capacity of 48 individual unique interactions with the ability to get up to 5 binding curves for each of them, producing 240 binding curves per cartridge. Sample volume, again, is 2 microliters per well, very minimal, which is nice. Crude sample compatible, so you don't have to worry about purifying your lysates or your serums. And the association-dissociation range fits most of the range of traditional SPR instruments, allowing for KD determinations from micromolar down to picomolar. We have a wide variety of applications available, such as kinetics, screening, quantitation, epitope mapping, and binning, and it's compatible with a wide variety of different biomolecules, such as your nutritional proteins, antibodies, peptides, nucleic acids, and so on. We have various different sensor chemistries depending on the needs for your research. Now, where is SPR used and why would you need to use it? So SPR is a gold standard technique because it's used throughout the drug discovery process at every stage. Our products are such a powerful platform for proteomics because they're extremely versatile. They have a critical role to play and unique advantages of every step of the biological discovery process. It's applicable to all platforms, including proteins and peptide therapies, antibodies, antibody drug conjugates, multispecifics, cell and gene therapies, oligos, and on and on and on. Alto provides critical data throughout each stage of the drug discovery process, from target ID to bioprocess QC, and spans all therapeutic areas, including oncology, immunology, infectious diseases, and more. The Alto is such a powerful platform for proteomics because it's extremely versatile, and as mentioned, plays a critical role at each of these steps. In the early stages of hit identification and screening, Alto enables scientists to quickly screen hundreds of thousands of samples to find that needle in the haystack. Alto is then able to find your hits. After you find your hits, is able to then quantify them, which it can do with its large dynamic range and excellent sensitivity. And then once these best hits and cell lines are identified, you might need to rank those to determine which ones have the best binding binding profiles based off their binding kinetics and affinities. And then lastly, it's also critical for scientists to understand where on their targets, if they're working with antibodies, that they are binding, which we can do through epitope binding and mapping. Next, I'm going to go through a few case studies of binding kinetics for some challenging transmembrane proteins. First, why do we feel the need to study these transmembrane proteins? Now, what a transmembrane protein is a protein that spans the lipid bilayer of a cell, typically has a few transmembrane components and some extracellular domain loops. Now, abnormalities in the function of these transmembrane proteins have been found to be associated with a number of diseases, which has made them a really interesting and unique drug target, with approximately 50% of drugs targeting these transmembrane proteins. Now, due to the complexity of these transmembrane proteins, to develop effective therapeutics against them, a full-length and biologically relevant form of these proteins needs to be isolated. Traditionally, these multi-pass transmembrane proteins are very difficult to work with or to purify due to their complex structures with multiple hydrophobic transmembrane regions. They also typically exhibit low expression levels and in vitro insolubility, or have unstable activity after purification. Because of this, we're working with ACROBiosystems that have enabled the use of these transmembrane proteins in various different platforms to stabilize them, such as micelles, nanodiscs, or VLP formats. In this first example here, we're going to talk about some interaction studies we did with antibodies binding to the claudin 18.2. As mentioned, many human diseases are known to be associated with these transmembrane proteins, making them ideal candidates for drug discovery. These claudins are a family of proteins that form tight junctions and recently have been identified to assist in cancer prognosis. The overexpression and inappropriate localization of the claudin 18.2 makes it an attractive target for therapeutic intervention. In this study, we were able to get SPR-derived kinetics for a human claudin 18.2 protein in a nanodisc, binding to various different human anti-claudin antibodies. This was performed by creating an anti-IgG capture surface, capturing our antibody on the surface, and then flowing various concentrations of the nanodisc and protein over the sensor surface to acquire these binding curves. Because of the high quality reagents in the nanodisc format, we were able to get high affinity or high quality binding kinetics to show the high affinity of this interaction, which matched very well with other traditional SPR characterizations of it. In the second study, we were determining kinetics against the CD20 with bispecifics against the CD20 antigen. The CD20 is a protein that is found on most B cells. Now, it is also found in higher than normal amounts in patients that have certain type of B cell lymphomas and leukemias, making it an ideal target for therapeutics. More recently, bispecific antibody therapies have been approved that target both the CD20 on the surface of these B cells, as well as the CD3 on the surface of T cells, to act as a bridge to keep the T cell in close contact with the cancer cell. Now, the CD20 reagents are typically available as full-length recombinant proteins or only recombinant proteins of the extracellular domain. Due to the low expression titer and unstable bioactivity, it is difficult to find active CD20 proteins on the market, and thus is why we've turned to ACROBiosystems to develop this assay with a full-length CD20 transmembrane protein in cells. And we'll see the reason for that in a moment. We initially started this study by trying to determine kinetics of a bispecific to a CD20 using these recombinant extracellular domain proteins. As we can see in these two examples here, we had flatline or no binding data present. What we did next is we used the CD20 protein produced in the nanodisc platform from ACRO. And we were able to get very nice binding curves to characterize this interaction. We characterize this a few ways with a traditional multi-cycle kinetic interaction, showing the 5 different binding curves on the right-hand side here, as well as doing a quicker single cycle kinetic titration series. So the bottom left figure that is shown here. In this situation here, we simply titrated from low to high concentration of analyte to determine our binding characterizations. We were able to get very close data to that was found in literature. This also highlights the need of not only, ensuring that the assay is designed correctly, but ensuring that the reagents that you use meet the quality that you need for your interaction. In this case, needing a fully formed transmembrane protein in a state that mimics its native function. So that is all that I have for you today. Hopefully you've gained some insight into how Alto can help your binding studies by allowing you to save your precious sample. Again, only having to use 2 microliters of your material allows you to save your time and your money. It allows you to also maximize your data quality as well as your time with the automated on-chip dilutions, and hopefully can simplify your discovery with our pre-designed templates and our automated analysis in the portal. At this point here, if you have any questions, please feel free to put them into the Q&A and we'll get back to you at the end of the webinar. And I will now pass it back to Anil.
Hello. Thank you, Mike, for the presentation. Now I will take over and I will be presenting from my side, which will have the contents. In the beginning, I will give an introduction about our company, ACROBiosystems, and then take you through the transmembrane protein challenges. And later on, I will also showcase the FLAG full-length active gallery, which is a technological advancement that ACROBio has achieved. And in the end, I will take you through some of the case studies with some of the challenging membrane proteins. So here, ACROBiosystems is a multinational company with more than 800 plus employees, and we have facilities in 12 different locations in 12 different international locations. We are covering more than 70% 70 plus company locations. And the company has a long history of strong collaborative partnerships with leading pharmaceutical companies like Pfizer, Novartis, and Johnson & Johnson, along with top academic institutions. ACROBiosystems is not just a single company. It is actually having subsidiaries like Biocidin, Condensed Capital, AcroDiagnostic, all focusing on innovative biopharmaceutical and health product and business models. So here in this slide, you can see different locations where our company is based. For example, Shanghai and Beijing, where our manufacturing facilities are. And in Basel and Newark, we have logistics center, which are catering to European Union region, as well as in the US region. In London, Boston, and St. Jose, we are having dedicated teams which are collaborating directly with the leading companies and engaging with them in our collaborative projects. Membrane proteins are essential components of cellular membranes involved in critical functions such as signal transduction, transport, cell-cell recognition, They are the targets for over 60% of approved therapeutic drugs, addressing conditions like hypertension, cancer, neurological disorders. Abnormalities in these proteins can lead to various diseases, making them crucial for developing effective therapies. Their role, particularly in enzyme activity, signal transduction, transport, and cell adhesion, underscore their therapeutic relevance. A category of membrane protein are checkpoint proteins, which are membrane proteins primarily found on the surface of immune cells. Key examples include PD-1, which are programmed cell death proteins, CTLA-4, which are cytotoxic T-cell lymphocyte-associated proteins, and also PD-L1. They have crucial role in cancer cell therapy development, where these checkpoint proteins are targeted using immune checkpoint inhibitors to enhance immune response against tumors. There are currently a large number of drugs in market which target these checkpoint protein, such as pembrolizumab, nivolumab, which are highly successful and widely used drugs in the market currently. Multipass proteins are primarily the key targets for the drug development, and they constitute over 70% of total transmembrane proteins. These transmembrane proteins have multiple transmembrane domains, and however, producing and purifying such kind of high value target proteins is challenging because there are multiple aspects of development, production and stabilization of these proteins. The key reasons for this is that their structure is quite complex, and in terms of solubility and stability, there are a multitude of challenges. Due to this reason, there have been several side approaches of generating relevant target domains of the therapeutically relevant membrane proteins. This include using peptides or simply extracellular domains, or using whole cell or even using whole cells. This approach has limited success because small domains are usually not stable and do not attain even the physiologically relevant conformation. Therefore, the antibodies raised against such proteins may or may not succeed in the later phases of therapy development. It is therefore inevitable and logical to produce and stabilize full-length membrane proteins. However, the key challenges like low expression levels, low abundance due to toxicity during overexpression, and solubility and stability issues in the form of aggregation need to be circumvented. ACRO has recently achieved huge technological advancements and have identified innovative techniques to express, purify, and stabilize these full-length proteins by improving expression systems as well as the expression levels, and through different novel stabilization technologies. We have named it as full-length active gallery, and in the subsequent slide, I will showcase you this technological advancement from our company. So we have stabilized the key membrane proteins in 3 different formats, which are in detergent micelle form, in nanodisk format, as well as in VLP format. These formats are serving as a platform for stabilizing many challenging full-length membrane proteins of high therapeutic relevance, and are amenable for immunization as well as analytics development using ELISA, SPR, and BLI-based technologies. In the upcoming slides, I will explain in detail these individual platforms, taking examples of full-length membrane proteins of high therapeutic relevance like CD20 and Claudin 18. Here I will showcase the 3 platforms and showcase the solubilization and stabilization of CD20 protein, which is a multi-pass protein and a prominent target for anti-cancer therapy development. In detergent micelle format, The membrane protein is wrapped in a hydrophobic detergent environment, such as the key epitope region relevant for the antibody screening are still exposed and the molecule retained its physiological conformation. We screen for multiple expression hosts and detergents to identify most stable format. Here you can see the stabilized CD20 has been used as an antigen in ELISA and SPR applications to showcase the stability and conformational integrity. The IC50 and the KD values shown here conform to the literature values for the commercialized rituximab and ofatumumab antibodies. Here in this slide, CD20 protein in VLP has been presented. VLP is a virus-like particle and is a type of recombinant protein that mimics the structure of a virus, but is not infectious because it lacks any viral genetic material. These particles are used to present membrane proteins in a more natural lipid environment. The CD20 VLP has been demonstrated here to showcase that the key epitope region relevant for the antibody screening are still exposed, and the molecule retain its physiological conformation. Since VLP can even have multiple number of molecules embedded into the same core, they are most ideal for immunization but less so for analytical development. Membrane nanodiscs are nanoscale disc-shaped lipid bilayers that are stabilized by surrounding scaffold proteins. Typically, these are derived from high-density lipoproteins, and these nanodiscs provide a more native-like lipid environments for membrane proteins, allowing them to maintain the structure and function outside of the cellular context. Membrane nanodiscs provide a natural lipid bilayer environment which is essential for maintaining its conformation, stability, as well as biological activity of these proteins. And these membrane nanodiscs are often more stable than those of the detergent micelle, which I presented in the previous slide, reducing the risk of any denaturation. Therefore, the proteins in nanodisc enable the functional reconstitution of these membrane proteins, allowing for detailed activity assays and kinetic studies. In summary, I want to highlight that the flag platform from ACROBio is a tremendous leap, and it has enhanced our capability to solubilize and stabilize any full-length multipass membrane protein and to obtain them in functionally active conformation for any downstream applications like immunizations or analytical development. Here I want to summarize that detergent micelle format and nanodisc formats are most amenable for analytics development, whereas VLP configuration for the multiple embedded transmembrane proteins molecules is most suitable for immunization purpose. Apart from the stabilization of protein, at ACRO we ensure generation of a very good quality control data package to ensure purity, uniformity, and bioactivity. Here you can see for the purity a regular STS page for assessing the uniformity. We do a DLS or SEC-MALS based assessments, and also for the bioactivity, these are mainly the analytics assay, for example, SPR, BLI, and ELISA based assays that we conduct routinely. We employ not just regular STS, SEC-MALS, and bioactivity assays, but are open to include additional QC assay methods such as DLS, EM, which is electron microscopy, and it is our highest priority to ensure batch-to-batch consistency. In the field, actually, for membrane proteins production, batch-to-batch consistency is a major concern, and that's why at ACRO in our quality management system, a lot of stress we are putting in this direction that our batches that we produce for such membrane proteins are consistent. This is a showcase slide to highlight that we have multitude of proteins that are already at our catalog, which can be directly purchased by the clients. For example, not just CD20 and Claudin 18.2, which I showed here, but also CD133, GPCR proteins, and different kind of chemokine receptors They are also available in all the 3 different formats that I showed from our platform, like detergent micelle, nanodisk, or VLP formats. And there is continuous development of membrane proteins from different therapy areas, for example, metabolic disease, autoimmune disease, neurological disease, rare diseases, which are highlighted here, these proteins from the different categories. So at ACRO, we are in a continuous process of development of these membrane proteins in the 3 formats which I highlighted, which are amenable for both immunization as well as analytics development. In cases where some very unique membrane proteins are being pursued by the companies, and they see that they are finding it challenging to produce these proteins, we also offer custom production services employing our flag platform. So I encourage you to engage with us in this direction if you are finding it challenging with your protein. We have very good experience. More than 300 plus product development scientists with the PhD degrees are employed, and they are working in this direction. And here in the European Union region where our team is based, and also in US, we have multiple engagements with big pharmaceutical companies where we have delivered to them in a collaborative way multitude of these membrane proteins for their downstream applications like immunizations, analytical assay development. So feel free to connect with us in this direction, and we are happy to support you for your therapy development. So this was my last slide, and I'm happy to take up the questions.
Anil, Mike, thank you so much for your informative presentations. And yes, it is now time to start our live Q&A portion of our webinar. Now, we have lots of questions that have already come in, but to our audience, if you have any questions and you haven't submitted them yet, please do so now. Type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. But before we dive into that live Q&A, I want to bring your attention to the far left of your screen. We have some survey questions for you, and your participation is greatly appreciated. So thanks, Mike and Anil. Let's dive in. All right. Let's start with this question. Mike, I believe this one's for you. If we want to use SPR for transmembranes, is there any special sample prep needed for the study of transmembrane proteins, especially multipass proteins?
Yeah, that's a great question. I would say with these transmembrane proteins, as long as you have them in some form of stable conformation, so I mean, presumably they probably wouldn't be stable in just aqueous solution, but again, using like a micelle or a bicelle or a nanodisk or a VLP, then you would just use them as you would any other biomolecule on the instrument. So in that nanodisk format, for instance, as we used in these studies, we would just have them in our standard PBST buffer. We found the tween 20 concentration was fine, and it did not cause any denaturation or breakup of the nanodisk. And then you could use it as either the ligand or the analyte in the assay. If your transmembrane protein has a tag, like a his tag or something of that nature, we could capture it via that tag and use that as the ligand. Or if you want to use it as the analyte at various different concentrations to get your binding curves, it's also suitable in that format too.
Thank you, Mike. Anil, let's jump over to you for this next question. It's 2 parts.
Yes, sure.
How do you handle variations between different batches of recombinant proteins? And do you collect batch consistency data?
Yes, so in my presentation, I have highlighted and put a stress on this point. So current challenge with membrane protein is the batch-to-batch consistency. In ACRO, we are actually putting a lot of stress on that. And that's like our quality management system. In this direction, I put thresholds on batch releases. If they do not conform to those quality standards and conforming with the previous batch, if the data is deviating, then we are not releasing that batch. So a number of analytical methods, as I highlighted, DLS, SEC-MALS, based assays that we are employing to make sure that we are assessing accurately and putting stringent thresholds on the batch releases.
Thank you, Anil. Mike, I'm going to jump back to you. For nanodisk TP, what mass do you use as the immobilized ligand?
Yeah, that's a good question. So the the mass that we would use would be dependent on the interaction that we're studying. And by that, I mean the molecular weight of the transmembrane protein and nanodisk and the molecular weight of the analytes. In a typical kinetic assay, we want to keep that mass as low as possible, where we can still detect an analyte binding signal. This way we can help prevent stuff like mass transport limitation or rebinding. If we were to, for instance, capture that transmembrane liquid via its tag, we would probably start somewhere in the neighborhood of like 1 to 10 micrograms per mil. If we were to try to amine couple it via primary amine onto a carboxyl surface, we would probably go anywhere between 5 and 25 micrograms per mil. But again, the density on the surface would be determined via the sizes of the molecules we're studying.
Thank you. Our next question. Hi, thank you for the presentation on Alto Digital SPR. I'm wondering how easy it is to test out different chips. There's a few questions here. Are they embedded in the cartridge? Also, since the buffer is part of the cartridge, how do you test out different buffer conditions? Thank you.
Yeah, that's a great question. We have quite a few different, what we would call optimization protocols that we could run on there. So each cartridge has 16 individually addressable channels. So you can think of it, I guess, as having 16 unique chips on there, so to speak. What we could do is we could use a select few conditions in each channel. So for instance, if we're trying to test out different immobilization densities, we could go low, medium, high on different channels. Also, each channel has its own individually addressable buffer well. So on the cartridge, we have 8 buffer wells that we can load. So in effect, you could try 8 different buffers, 1 per what we call lane. So we treat a pair of sensors as a lane. And that way you can try quite a few different conditions on one chip. So there's quite a bit of flexibility there.
Thanks, Mike. Now, Anil, I'm going to jump back to you again.
Mm-hmm.
How do you ensure and validate the functional activity of your recombinant membrane proteins?
So this is a critical point, basically. Like, once you embed these molecules, whether it's in nanodisk format or in micelle format, they must conform to their activity profile. And for this, basically, we use functional assays like ELISA and SPR-based analytics methods. So this is what we are usually using. And there are already, if existing antibodies are there, I gave an example of in my presentation, for example, rituximab against CD20. So we directly use those antibodies to see whether those epitopic regions are exposed and proper conformation is attained. And so this is how we basically check for. And also, I have touched in my presentation also DLS, dynamic light scattering, just to see the monodispersity of these molecules, EM method also to see whether they are homogeneous. So this is how we ensure their overall profile and activity in terms of the analytical method later on.
Thank you, Anil. Now, is there any experience using VLP for SPR on the Alto?
Yes, I can take that one. There is actually. We've had quite a few customers use VLPs on the Alto. The reason why we didn't showcase some of that here was, I think this is going to answer another question that was asked here either also. But on the VLP, we'll have multiple subunits of that protein on there. So if we were to use it as an analyte, we wouldn't be able to fit it to a proper binding model. This is why we like to stick to the formation in the nanodisk, because we know it's a 1-to-1. But we have had great success in the VLP. We've used it also as the ligand, where we've captured it, say like an anti-target specific antibody capture surface. So it is feasible to use both methods.
Yeah, I just want to add what Mike just mentioned, because these 3 formats that I presented, like nanodisk and micelle format, there, you can control the number of molecules that are embedded in the micelle and nanodisk. But in the VLP, there is a challenge, because we can, even at ACRO, we cannot ensure that, 1 or 2 or 3 molecules per VLP are embedded. This is something, a process which during the budding, that's how the core particle takes the molecule along. And as Mike rightfully mentioned, since there is a heterogeneity in the number of molecules present on the VLPs, good fitting model might be a challenge. And this is something we encounter and we recommend also that better use nanodisk or micelle formats, but VLPs can be more utilized in immunization purposes, where you can enhance basically the immunogen aspect of it by having multiple number of molecules.
Thank you both. Now, has acroantigen been used for tripartite AGAG FCR interactions scored by SPR, either by Alto or other platforms?
Yeah, that's a good question. Myself personally, like in our lab in our office, we have attempted a couple, like 1 or 2 tripartite experiments, and there's a high likelihood that the antigen that we immobilized was probably from acro. But in those instances, it would have been a standard recombinant protein. It wouldn't have been like a transmembrane protein. We've only ever done the ones in-house with, I think we might have done it with, I don't even know if it was like an anti-hemagglutinin protein or something like that, but it would have been a recombinant protein, not a transmembrane protein. But we have tried that, and we had some pretty good success with that too. I don't know if Anil, if you guys have ever done if you had any customers do any of those types of experiments where they load the antigen on the surface, add the antibody, and then the FC receptor to detect binding. I guess you probably wouldn't be aware of any. Okay.
So
I hope that's satisfied. Yeah. Sorry?
No, I didn't get the last sentence regarding the FC receptors embedding
yeah. So this is like a 3-part like assay where they load the antigen on a sensor surface, bind the antibody to it, and then they bind the FC receptor to the antibody. I don't know if you've had particular customers reach out for reagents for that, but.
So we do have a lot of customers who ask for FC receptors, but this sandwich kind of experiment, we do not sometimes know how they are planning to use it. But there are a lot of customers who ask for FC receptors like CD16, 32A, 32B, and CD64. And at ACRO, actually, we are specializing in generating those as recombinant proteins, not just as recombinant proteins, but as also VLPs, as stable cell lines, as reporter cell lines. So there's a lot going on in this direction as well, like, where people need to have ADCC and ADCPS development. And in the beginning, it has to go through the analytics development part. And as you rightfully mentioned, a sandwich assay is a possibility as well. Antigen and then the therapeutic antibody under development, and whether it's binding to any of the FC receptor of interest. So what you suggested is in principle possible and doable.
Great. Yeah. And if the question I was asked, if they have any, specific questions on design or analysis, please feel free to reach out to us.
Great. And that's a good point. I do want to mention that any questions we don't answer today, and additionally, during the on-demand period. So if you have questions, feel free to come back, watch the webinar on demand, submit your questions. They will be answered via the email address that you used at your time of registration. So today's not the only time you can submit questions. So let's jump in to our next question. How stable are the membrane proteins in the SPR experiment?
Yeah, that's a good question. So, we presume that they're quite stable since we get really nice, robust, and reproducible binding curves. We use, pretty like non-harsh conditions. We like standard aqueous buffers. There is a little bit of surfactant that's in there, which there have been some questions, does that affect like the nanodisk or the VLP? But in our studies, we've run assays that are quite a few hours long, and we don't see any deterioration in binding. So they seem to be quite stable in our environment.
Thanks, Nick. By using these methods and techniques, can we differentiate and functional test of the phosphorylated protein from its non-phosphorylated counterpart?
Yeah, I saw that question. I think I kind of understand it. It might require, please, I encourage you to follow up, email us. From an SPR method standpoint, I'll let Anil comment on this from other techniques that they use, but from an SPR standpoint, what we could test would be more so like, I guess, yes, no binding, as well as does the phosphorylated protein, this is like, post-translational modification affect the binding kinetics. That is certainly something that can be assessed on the SPR. It might be an instance where the phosphorylation enhances binding, non-phosphorylated counterpart has no binding. We would definitely be able to see that. But as for, if it affects, I don't know, like enzyme kinetic rates, something like that, I wouldn't be able to tell you on the SPR.
Yeah, I want to just jump in to say, like, sometimes for phosphorylated and non-phosphorylated variant of protein, there exist antibodies for a specific variant. So they can be utilized as well if those antibodies are available, and they can indeed be raised as well for a phosphorylated version and or non-phosphorylated version. So they can be employed in binding studies if they are available. It's a long shot, but just if somebody really is hell-bent to go in this direction, can generate those reagents and can also identify and perform those experiments.
Thank you both. And I do want to mention, as I said, you can come back, watch on demand, and submit questions. But additionally, info@nicoyalife.com, and that's NICOYALIFE.com. You can submit your questions there as well. So you have two options. And we are at time for questions. Mike, Anil, I'd love to give you an opportunity to give our audience some final comments.
Yeah, sure. Yeah, I'll go first, I guess, Daniel. Just really appreciate all your time today for joining us. Hopefully we've provided some insight into how both our platform and ACRO's reagents can help in your transmembrane research. Looking forward to hopefully working with some of you in the future.
I also want to summarize what I actually presented, and I find it interesting that the field of membrane protein, it is a target for more than 70% of the pharmaceutical that we are developing currently. And there has been a challenge targeting these proteins through antibodies or other aspects. But now it's not a challenge anymore because more and more proteins being stabilized and being made available. And as I presented also these three formats from the FLAG platform, we are making it possible for our clients that if they are working with these membrane proteins, the challenge on solubilization and stabilization is not their problem anymore. They can deal with it and they can ask us and we can support their therapy development. We can provide these proteins to them. And that's why my last slide was in this direction for custom development. Some of the novel proteins they are working with, they can join hands with us. And we have timelines of 8 to 12 weeks when we are delivering these proteins. So, nothing can stop you from pursuing your membrane proteins. And as Mike has showcased, the analytics development with our proteins, that this is also not anymore a bottleneck. So this device that Mike has presented is also quite useful in assessing the binding affinities and characterization of the antibody candidates that we will be pursuing in the future.
I do want to extend a big thank you to both of you, Dr. Michael Piazza and Dr. Anil Kumar, for your time today and important research here. I also want to thank LabRoots and our sponsor, Nicoya, for underwriting today's educational webcast. Now, before we go, I do want to thank our audience as well for joining us and for their interesting questions. We had quite a few questions we were unable to answer. So as I mentioned, these questions and those that are submitted during the on-demand period, and additionally, to info@nicoyalife.com. They will be answered via the contact information you provided at registration or replying to your email at info.nicoyalife.com. So today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Now, if you missed filling out that survey, it's going to pop up on your screen in just a moment. And to all our friends out there, thank you again for joining us. Have a great day, everyone. Bye-bye.
Thank you, everyone.